Hello everyone, my name is Xu Yisheng from Electrical Engineering in Yangming Chiaotong University. And today's my presentation is about more than double the fun with two-photon excitation microscopy. And here is the article which published in uh, Communication Biology. So we can first discuss why two-photon microscopy is important. Conventional single-photon fluorescing microscopes lose constraints, resolution, and sample viability once we image deeper than about 100 micrometers. Because visible light scatters, excites fluorophores all along its path, the rapidly photobleach tissue. In 1990, Dank and Webb resolved that bottle tech with two-photon excitation. A femtosecond near-field red laser concentrates photon densities only at the focal point, where two low-energy photons are absorbed simultaneously. That nonlinear process gives us intricate optical sectioning, 3 to 5-fold lower photography and penetrate depths that routinely exceed 800 micrometers in living brain. The paired brain slice image on this slide constrains the heavy, shallow, single photon view with a script millimeter scale two-photons view, setting the stage for everything that follows. Let us unpack the mechanism in 60 seconds. On the Jablonski diagram, a green fluorophore jumps from the ground singlet S0 to the excited singlet S1, only when it absorbs two 900 nanometers photons within the femtosecond window. Because fluorescence intensity scales with the square of the instantaneous power, excitation is confined to a sub-millimeter focal volume and vanishes elsewhere. Near-infrared light also scatters an order of magnitude less than blue or green light, so we reach deeper with a higher signal-to-noise ratio. Keep three phrases in mind. Nonlinear power squared depends zero out of focus excitation and lower scattering near IR photons. They are the physical pillars of all modern two-photon instruments. Moving on the standard optical path. Here is a canonical setup, a locked laser, and use some specific scan mirror, scan the tube length and a high NA water immersion objective from the familiar confocal-like path, but we drop the pinhole instead. Large area photomultiplier tubes sit right behind the objective in the so-called non-discrete detection arm. They capture every scattered photon that exits the tissue, boosting collection efficiency by roughly a factor of 3 compared with pinhole limit discrete detection. Remember this architecture because the next 3 slides are all extent of this backbone. And the first tips that we will introduce is the wider, the two-photon light shift copy. The two-photon pioneer quickly realized that point gain is lower and inherently narrow. Enter light sheet fluorescence microscopy. A thin laser sheet illuminates the sample from the wide while a camera collects an entire plane at once. The digitally scanned two-photon light sheet shown here widens the field of view to 400 by 800 by 250 micrometers and acquire whole volumes in half a second while keeping the photon density high enough for nonlinear excitation. Because only the in-focus plane is illuminated, the energy dose per drops by 5 order of magnitude, which is where we can record an entire zebrafish brain or mouse cortex for a minute without thermal damage. Next one is faster. Speed, not width, is the next bottleneck. Two-photon light-field microscopic insert a micro-lens array in front of the camera, capture the angular information in a single shot. Computational reconstraint then deliver three-dimensional volume at kilohertz rate, faster enough to resolve some specific cells that only have in a few milliseconds. The trade-off is a two-fold loss in spatial resolution, but the benefit is an unprecedented ability to image hundreds of neurons forming in real time in an intact, scattering brain. In other words, it does for two-photon what high-speed photography did for physiology. It shows events that standard point scanning would completely miss. Next one is the deeper. Depth eventually hits the scatter wall again, so we physically move an optics, a sub-military gradient index or GRIND, lens implanted through cortex into the hippocampus, the excitation beam, and returning fluorescence to a compact skin head. Mount that head on a freely behaved mouse and we can monitor place cell activity, dopamine release, or social interaction circuit more than 1 millimeter below the brain surface, territory unreachable by any other optical techniques. The entire microscope weighs under 3 grams. Keep the animal's behavior natural while deliver single-cell two-photon resolution. Two-photon is not limited to structural image. When we couple it to a single photon counting electronics, we open quantitatively doors. Fluorescence correlation scopy records nanosecond scale intensity fluctuation at 14 kilocount per second. Autocorrelation curves that yield diffusion coefficient and binding kinetics inside living tissue. Forcing's lifetime image collects decays profile and maps free versus protein-bounded NADH. Give a label-free readout of cellular metabolism up to 700 micrometers deep. Combining both techniques on one microscope let us measure in the same cell how molecules move and how much ATP the cell is burning. Signal is useless if the specimen dies. Photon damage depends on both average power and peak power squared. Keep the average below roughly 16 milliwatts at the back pupil while shortening the pulse width to 100 femtoseconds and dropping the repetition rate from 18 MHz to a few MHz. And let peak intensity, great for two-photon efficiency, yet leaves the mean thermal loads unchanged. Line scanning or light sheet strategies further spread the dust, allowing 2-minute time-lapse recordings of hippocampal slice with no observable swelling or bleach. To wrap up, remember 3 words: wider, faster, and deeper. Two-photon light sheet give us panoramic volumes with negligible phototoxicity. Light field system push the temporal resolution into the kilohertz domain. Green endoscopic breaks the depth barrier and follows narrow circuit in freely moved animals. Photon counting turns the same platform into a biochemical analyzer, and careful control of pulse parameters and polarization keeps our sample alive. Open source, hardware redshift probes, and AI reconstruction stand ready to demonstrate and excite those tools. Thank you for your attention, and I am happy to take your question now.